Hey everybody! In the last video, you got a brief introduction into the basics of genetics. However, before you move on, you have to learn the language a little bit. So in this video, I'll be teaching you the most commonly used terms of the field, how they relate to each other, and using them to describe the underlying cause of a genetic disease, sickle cell anemia. Remember to fill out the learning guide as you go. We'll be answering 2 questions in this video. Okay, so question the first. What is the meaning of each term used in this video? And question second, how do these terms relate to each other? Okay, so put that up there. And the terms that I'm going to use in this video are going to mostly be in black. So that'll cue you to know which terms to focus on. Also, keep in mind that you should be able to define each term in your own words. Okay, to review, a gene is a length of DNA that encodes a protein, and DNA is made up of 4 different types of nucleotides, T, A, G, and C. And DNA is packaged through various means into a chromosome. So now we can start to relate these terms to each other, and we'll start with DNA. DNA is made of nucleotides. And there are 4 types of nucleotides, which are, of course, A, T, C, and G. And DNA is packaged into a chromosome. And of course, a gene is just DNA that encodes a protein. And there are many genes in the human genome. We'll consider one in this video. Our example will be the HBB gene from humans, which is important for red blood cell function. Keep in mind that a chromosome can contain millions of nucleotides and dozens and even hundreds of different genes. This is only part of one chromosome being shown here, and you can see many different genes on it. And if we want to refer to all the DNA in the cell or all the DNA of a given species, the word we use is genome. So a cell or a species' genome is just all of its DNA. And in the human genome, there are about 3 billion nucleotides organized into 23 different types of chromosomes. So different genes can control different traits. And again, the gene that we're considering here is the HBB gene, and the trait that it controls is red blood cell shape. And next we're going to see how it does this. Now, of course, genes don't control traits directly because DNA doesn't really itself do much. They control traits through the protein that a gene encodes, or in other words, provides instructions for how to make it. Now, the first step on the way to protein production is when our HBB gene undergoes transcription, producing an RNA molecule, which is similar to DNA, and the RNA copy of DNA also has the same instructions that the DNA has for how to make or encode a protein. And now RNA is read during translation, and this is the process that actually produces a protein. And it's the protein itself, not the gene that encodes it, which carries out some cellular function. The HBB gene encodes a protein called beta globin, which is needed for red blood cells to carry oxygen from the lungs to other parts of the body. So the function of the beta globin protein encoded by the HBB gene is in oxygen transport. However, sometimes we'll just say that the function of a gene, in this case the function of the HBB gene, is an oxygen transport, always with the understanding that it's actually the protein that's doing the work. Now, a protein's function, in addition to acting at the cellular level, can also control a trait. And a trait is just an observable characteristic of an organism. And we said before that an example of a trait is red blood cell shape, but other traits that might be easier to think about include something like height, hair color, as we saw in the previous video, as well as eye color. In a minute, you'll see how this protein's cellular function can control the trait of red blood cell shape. And we'll talk more about this later. Sometimes a gene's DNA can be changed by a mutation. A mutation changes the sequence of nucleotides of a gene in some way. Here, it's been changed from an A to a G. This creates a new version of the gene, and this new version is called an allele. And we can call, for example, the original version of a gene the HBB plus allele of the HBB gene. Likewise, we can call the new allele HBBS. So HBB plus and HBBS are two different versions or alleles of the same gene, which is HBB. This new HBBS allele, when transcribed and translated, can encode a slightly different protein that carries out an altered function than the original version of this protein. Here, this new allele produces a slightly different protein that doesn't transport oxygen very well. So here we can see how a mutation which generates a new allele can lead to altered protein function and ultimately, as we'll see next, to genetic disease. So we have two alleles of our HBB gene, HBB plus, and then through a mutation, a second allele we've called HBBS. The HBB plus allele encodes normal beta globin protein, which is fully functional. The HBBS allele encodes an altered version of this protein, which has altered function. The HBB plus allele, because it encodes a normal functional protein, gives normal, round, red blood cells. And so this is the phenotype for this allele. The phenotype for the HBB plus allele is round. The HBBS allele, shown here, and its abnormal protein that it encodes causes red blood cells to adopt a sickle-like shape. These two different appearances of red blood cells caused by two different alleles, these two different appearances are called phenotypes. And a phenotype is just a version of a trait. Here, the trait is red blood cell shape, with the possible phenotypes of this trait being either round or sickle-shaped. These sickle-shaped cells die prematurely and result in reduced oxygen transport if there are too many of them. This results in anemia, a genetic disease caused ultimately by the mutation that changed the HBB plus allele into the HBBS allele. Humans and many other organisms have two copies of each gene. In other words, they have two alleles of each gene. Which alleles an individual has is known as that individual's genotype. If a person has two of the same allele, they are called homozygous. For example, HBB plus is a homozygous genotype. If an individual has two different alleles, they are called heterozygous. For example, HBB plus HBBS is a heterozygous genotype. Here, since we're considering only two alleles of a gene, that means there are three possible genotypes: HBB plus HBB plus, HBB plus HBBS, and HBBS HBBS. The genotype determines the phenotype. Here, the HBB plus genotype gives all normal red blood cells, which is normal. The heterozygous HBB plus HBBS genotype produces both types of cells. But this still results in basically a normal medical outcome because there are enough normal red blood cells to transport oxygen. However, the homozygous HBBS genotype results in many sickle cells, which in turn causes the disease sickle cell anemia. So here we've seen how a mutation that can create a new allele causes that allele to encode a protein with reduced or lack of function, resulting in a phenotype that can lead to a genetic disease, sickle cell anemia. Keep in mind, though, that mutations that create a new allele which reduces or destroys the function of a protein are thankfully pretty rare. In fact, most mutations don't do very much at all. And part of the reason for this is that most of our genome, the human genome, isn't actually made up of protein-coding genes, it's made up of other DNA. And while harmful mutations are pretty rare, occasionally a mutation can occur that can actually improve the function of a protein or sometimes even add a new function to an encoded protein. If these beneficial mutations help an individual reproduce more often, this is how natural selection occurs. In other words, the new beneficial allele will be selected for over time. This is how evolution happens, and this will be the topic of our next video. Okay, so at this point you should be able to define all the terms that we've gone through in this video and understand how they relate to each other. Now, if you can't, no worries, just watch all or part of the video again. Now, in the next video, we'll look at the role of genetics in evolution. And this is pretty important, as evolution is really the central theory of all of biology. It connects everything in biology to everything else. So I hope you guys look forward to that, and I'll see you later.